Third presenter, and this is Dr. Sarah Bust. Sarah earned a BS in biochemistry from Virginia Tech and a PhD in microbiology from the University of Virginia. Prior to joining the Wyoming Public Health Lab, she completed a CDC APHL Emerging Infectious Diseases Research Fellowship and worked as a research scientist at the Wadsworth Center and NYS Department of Health. She also completed a medical microbiology fellowship at the University of Nebraska Medical Center and Nebraska Public Health Laboratory. She currently holds an adjunct assistant professorship in the Department of Pathology and Microbiology at UNMC. Sarah has worked with diagnostics development in industry, academia, and government laboratories and has developed tests for detection of various bacterial and parasitic agents of disease. She has published journal articles and book chapters and given lectures on range and diverse topics related to clinical and public health laboratory science. Sarah is a member of the Association of Public Health Laboratories and the American Society for Microbiology, and she is certified in medical and public health microbiology through the American Board of Medical Microbiology.
Thanks, and actually this is only the second time I'm talking about microbial source tracking, so it's a little new to me as well. And I took the approach with this talk of focusing on basic concepts in molecular biology so that you guys can understand some of these methods that we're going to be talking about. And I'm also going to talk a little bit about targets of these methods, as well as the techniques themselves. And I'm going to focus on the techniques that we are familiar with at the Wyoming Public Health Laboratory. So this is going to take you way back, right? Or maybe not back at all, I don't know. So the central dogma of molecular biology is that DNA is transcribed into RNA, and RNA is translated into protein, right? So the DNA within a cell contains the information for the proteins that that cell will eventually make, okay? And we're going to talk about genes, we're going to talk about genomes. And so just so you understand, a genome contains all of the genetic information of a particular organism or virus, right? So it contains all the information that encodes all of the proteins that that organism will ever make. Whereas a gene contains the specific instructions for making a single RNA molecule or a single protein. And I don't know what I did. As you may know, the human genome is organized into 23 chromosome pairs, and bacterial genomes are much more simple. They tend to be circular and much, much smaller than the human genome. Viruses can be circular or linear. So you may be asking, why is all this important? Well, it gets to the fundamental concept of microbial source tracking, which is that bacteria and viruses can genetically adapt to their environment. So if you think about DNA encoding proteins, and proteins being used to kind of work on things in the environment, so if you're a bacteria living in the gut, you need to degrade enzymes that are produced there that might kill you or something like that. And the enzymes that are in a cow are going to be different from those in a human, and you might pick up mutations that make your proteins function a little bit better with the host that you're living in's proteins as well. Does that make sense? And so these mutations are passed on to the next generation, and over time, the bacteria present in the guts of all these different species are going to differ. And so the theory is that you can match the microbes from a polluted site to the animal source and then point to the origin of the fecal pollution. So you've heard a lot about some of these targets already. Microbial source tracking targets include various species of bacteria, including Bacteroides, E. coli that we've talked a lot about, streptococci, enterococcus, rhodococcus, bifidobacterium. They also include viruses, human viruses like enterovirus and adenovirus, but also viruses that infect the bacteria themselves, like the Bacteroides fragilis bacteriophage or the F-specific RNA colophage. What I'm going to focus on are going to be methods that look at Bacteroides and E. coli. And you were asking earlier over here about persistence in the environment. Well, these two organisms have a different ability to persist in the environment. And so that question is also going to be an organism-specific question because as an anaerobe, Bacteroides is going to die in the presence of oxygen and not persist. So a lot of people think for microbial source tracking purposes, it makes a better marker because it's less able to persist. And E. coli, I mean, you guys have been looking at E. coli in water as a marker of fecal pollution forever. So that one just sort of seems like a logical organism to look towards for microbial source tracking purposes. And we'll talk about that a little bit at the end, whether that makes sense or not. There's also a variety of methods that are used for microbial source tracking. And these include host-specific PCR, real-time PCR sequencing. We're going to talk about all of those in depth. And gene-specific PCR is also used, real-time PCR sequencing. People use methods including phage typing, total community analysis, as well as pulse field gel electrophoresis sequencing, antibiotic resistance analysis, rep PCR, and ribotyping. You've heard a little bit about the difference between library-dependent and library-independent methods. That's a hugely important difference because these library-dependent methods are going to require many, many samples to be looked at. You're going to have to sample the stool of the animals that you suspect and just create large libraries using those techniques. And we'll talk about that in detail in a second. So of these, the methods that we're going to really focus on are the host-specific PCR in terms of library independent, and then pulse field gel electrophoresis as well as sequencing in terms of library dependent. And you may notice I have sequencing on there as a library independent and a library dependent method, and that's sort of because it depends on how you're using it, and I'll explain that too. On the next slide, I just want to go again through kind of the components of a PCR reaction, because we're going to talk about a lot of variations of PCR, and it's going to be important to understand the components because those are what creates some of the variables. So she talked about primers, which were the short oligonucleotides that hybridize to opposite strands of DNA and flank the region of interest, right? Those are what give the PCR reaction its specificity. They also, to a certain extent, define the sensitivity of the reaction, but for sure, they define the specificity. You know, the rest of the stuff, DNA polymerase, the triphosphates, and the buffers and additives, these are just standard components that make the reaction work. Something that I kind of left out of this talk, but is extremely important, is the template DNA. And that's the DNA that starts the reaction off. So what you're extracting from your sample, from your water sample. Extremely important. And then words that I'm going to be using that you may hear and not know what they mean, amplicon or amplimer, they mean the same thing. It's basically your PCR product, that exponentially amplified product at the end of your PCR reaction, okay? So I went through that because I want to explain to you the difference of conventional versus real-time PCR. So conventional PCR, sometimes people call it endpoint PCR. And this is considered an open system. And what that means is after amplification, you need to open the tube in order to analyze what happened in that reaction and see if you have a product or not, okay? So there's a couple ways of doing that. The most common is via this agarose gel electrophoresis, where you separate the DNA by size. So in the first lane over there, it's just a ladder of various size pieces of DNA and then 2 PCR products. Smaller pieces will move faster through the gel. So the piece that's a little further down is just a little smaller than the other. And it's stained with ethidium bromide, which gets into DNA and fluoresces with the UV light. Another way to do it, which is much less common, is this bioanalyzer trace. And this is actually analyzing the same PCR products over there. So this is a way to make it an endpoint PCR quantifiable. And they're also basically separating the products by size. And just, you know, the faster they come off of this column, the smaller that the piece is going to be. And then the size of the peak corresponds to how much DNA is there, right? So this is important because it's different from real-time PCR, right? I'm going to tell you what that is in just a second. But basically, some of the drawbacks of this type of conventional PCR type process are that you have to open the tube. And that might not mean a lot to you, but what it means to me in the lab is that I'm risking contaminating the assay. Because in there, you just saw there's billions of pieces of that product. When you open the tube, you can aerosolize them. They can get somewhere they shouldn't and contaminate your next reaction. So you absolutely want to avoid that. And that's one of the risks with this type of process. Another problem with it is that it's not really quantifiable. You know, you can do it like here, and you can even estimate based on how bright your band is. But it's not the best way to get something that's quantified. So for that, people use real-time PCR. And there's a small difference between real-time PCR and conventional PCR, but it makes a huge difference in what you're getting. Okay, so there's many methods of doing it. The way that's most important to microbial source tracking is including an extra component in your PCR reaction, and that component is a fluorescently labeled probe. And the probe is very similar to the primers in that it binds specifically to your target site of interest. But what it will do is when it binds, it will fluoresce and give off light that you can detect. So something that's important about this is that it's a closed system. I'll show you what it looks like to analyze this data in just a second, but you don't have to open the tube to get your result. And another thing is this probe makes the reaction more specific. It's just an added level of insurance that you're looking at the right target or looking at the target you think you are, because it has to bind specifically to the amplified DNA. Okay, so this is just kind of outlining. This slide kind of stinks, actually. But what it's showing you is that, you know, we'll look at this in more detail, but basically in a real-time PCR, you are detecting the products as they are created, whereas an endpoint PCR, you have to wait till the end of the reaction and then look to see what's there. So this is a better slide. So this is looking at kind of what you would see a curve of a real-time PCR reaction. And we often have cycle number at the bottom, and that's literally the cycle number of the PCR that you are at. So it's pretty standard for a PCR to go anywhere from 35 to 45 cycles. And the units on the y-axis are just sort of arbitrary light units, because remember I told you that probe puts off light that can be detected. So when something's detected at a lower cycle number, it means there's more of that target DNA in the sample. So in theory, real-time PCR is always kind of quantitative. I can look at this and I can say, okay, there was more template DNA here than there was here. And it's supposed to work that every 3 cycle numbers is a log. So these are approximately 3 cycles apart. So these are approximately 10 logs apart, or 1 log apart each, if that makes sense. But it's not always precisely quantitative unless you include a sample at a known concentration. Okay, so you might have noticed that people abbreviate real-time PCR as qPCR instead of RT-PCR, and that's confusing, right? And the reason for the Q is because it means quantitative. The reason they don't call it RT-PCR is because there's another kind of PCR that is RT-PCR, and that's reverse transcriptase, where you use RNA as your starting material. So the RT was already taken. We call it qPCR. And so I just kind of wanted to walk you through how we would get a number at the end of a, how do we tell you that there's 4 gene copies in your sample, 4 gene copies per milliliter? So what you do is you run a dilution series of either a known quantity of bacterial or virus particles, whatever, or a known quantity of DNA. So in this example, you know, each of these different samples, I guess, were about 10-fold apart. So they started with 10,000 CFU per mil, 1,110. and these each have corresponding CT values. Now, how you get the CT value for the sample is you set a threshold level, right? So we set the threshold level, and where your sample crosses that threshold is the cycle that it became positive. So sometimes people will give you a CT. Well, that's the cycle in which your reaction became positive based on the threshold that was set. And in this example, you can see the negative control remains negative below this threshold that was set. And so what you would do is you would just make a simple plot of 10,000 CFU per mil at a CT value of 7, 1,000 at 10.5, and so on. And then you would go back to some basic math, calculate the slope of that line, plug in your unknown sample's CT value, and calculate the starting quantity of your target in that sample. And so in this one, if we had a CT of 15 in an unknown sample, and we plotted this, came up with the slope of the line, y equals mx plus b, and plugged in this 15, we would come up with a 76.5 CFU per mil value at the end of the day. So that's how it is quantified. So how do people use this qPCR for microbial source tracking? Well, a variety of different ways, I think, is what you guys have gotten so far, and that's what I'm going to continue to tell you. But what I've seen most commonly is the focus on the Bacteroides 16S rDNA. And there are a variety of different published and well-categorized assays that look at the 16S rDNA region. It's a both highly conserved and highly variable, if that makes sense, region that is nice to use for this purpose. So there's human-specific targets. The most well-known is that HF183 assay. And then there's an EPA-approved EPA-patented HUM-M2 assay. And I guess what I want you to take away from this slide is that there are a variety of real-time PCR assays out there that are well-characterized, and probably the best way to use them is to use them in conjunction with each other. But like everybody has been saying, you have to keep in mind what's in your specific region before you go just throwing all these targets into the mix. So, for example, if you wanted to see if humans were a source of pollution in your region, you might use a total marker target, so looking for all Bacteroides from all species of host animals, and then use that in conjunction with these human-specific targets. And the nice thing is, like I said, there's more than one target for these hosts. So you get more solid information when you combine them. Okay, so what's the plus and minus of using qPCR for microbial source tracking? Well, on the good side, it's a rapid technique. It's library and culture independent, and it's relatively inexpensive. It's a sensitive, specific, and quantitative method. Is it as sensitive and specific as you guys want it to be? Probably not. Nothing is ever as good as everybody wants it to be, but it is highly sensitive and specific. There's many validated targets to choose from, and they have well-controlled assays. What's some of the downsides? You can have false negatives. So some things that can cause false negatives are with sample collection. You can collect too few samples. There can be dilution or target degradation. There can also be inhibition. Now, inhibition is something that we can control for in the reaction. There's a variety of things that will inhibit a PCR reaction, but what we do in a well-controlled assay is you can spike your sample with DNA, and you can amplify that from the sample to ensure that there's nothing in there that's inhibiting the PCR from working. So that's a component of a well-controlled assay, and that should be a component of the assays that you guys are using. There can also be false positives with PCR. False positives can come from contamination. Like I said, that amplicon, there's a lot of it, and if it gets into the wrong place, it can contaminate a specimen. But if you're using the right laboratory, they should have processes in place to prevent that. So at the public health lab, we're a clinical reference lab, so we are regulated by federal entities, and we are required to have certain processes in place when we run molecular assays. For example, we have to have a unidirectional workflow, meaning that we extract our sample in one location, we set up the reaction in another location, and carry out the reaction in a completely separate location, so that we are not creating opportunities for cross-contamination within our laboratory. There's also controls that you can run in the reaction to check for contamination. Everybody should be running a negative control every time they do a PCR to make sure the reagents are not contaminated themselves. Another thing about this kind of surprised me when I started doing some reading about microbial source tracking, but it makes a lot of sense, is that these real-time PCR assays cannot be used reliably to correlate the percent contribution at your source from each host species. Okay, so what I mean is, let's say in your sample, you have 100 CFUs per mil of Bacteroides. And of those, you get also a hit on a human target, and that comes up at 50 CFUs per mil. And then you get, another 50 from cattle or something. So in your sample, that may be what's going on, but in your watershed, there's things that are causing that to kind of be misleading. You can't say that 50% human and 50% cattle are contaminating, because there could be dilution, there could be degradation. You're looking at a specific species of bacteria, like Bacteroides, which may be present in animal feces at a different concentration. So I think that's a downside that surprised me, but it makes a lot of sense, actually. Okay, so what can we, or will we, is the correct word, be able to do at the Wyoming Public Health Lab in terms of real-time PCR? So we perform real-time PCR every day at the lab. We do it for pathogens, influenza, pertussis, norovirus, things like that. We have not used real-time PCR for microbial source tracking purposes. However, shout out to Paposia, we have begun looking at the microbial source tracking assays. We've gotten samples from Paposia, we've filtered them, we've extracted DNA, and we have purchased all the reagents to begin using an all Bacteroides assay, as well as a human-specific HF183, and potentially a human-specific HUM-M2, only that is patented by the EPA, so we're waiting to hear if we're going to get a license to be able to use that one or not. So we have the equipment, we have the reagents, and we have the knowledge on hand, but we haven't yet run this assay. We plan to do it within the month, yeah.
So it's basically just for humans, is that what you have?
So far, those are the reagents we've bought, and that's partially because we were working with Paposia, and that's what they wanted to look at. So we looked into the cow too from the beginning. We kind of outlined what we would need to purchase in order to conduct some of those assays. And when I asked for the HUM-M2 license from the EPA, I asked for a couple other licenses as well, just because why not? But yeah, right now that's what we have on hand. But like I said, we haven't used it yet. And part of that is we're strapped. We have limited staff, limited resources, and we frankly have other priorities that currently pay the bills. So we want to help. We think we could help, and it's just going to take some time. And so once we have these assays validated, I think it would be something that we can easily do. It's just we're not quite there yet. Okay, so I want to also talk about pulse field gel electrophoresis, because you guys have heard about this. And so I kind of want to take you through what that is. So basically, it's a type of genetic fingerprinting. It is a culture-dependent method, library-dependent method. It requires a pure culture of a known organism. In fact, you have to have one colony to start with. It's qualitative, but it's robust, and it's highly reproducible. There's very good discrimination with pulse field gel electrophoresis. And right now, the primary purpose of PFGE, at least in our laboratory, is to detect and track food and waterborne outbreaks of bacterial diseases. This is a historically proven method. It's been in use for over 20 years. We'll talk more about that in just a second. And it does allow, in some respect, for real-time communication between various groups. And we have Wanda in the back, who is a nationally recognized expert in pulse field gel electrophoresis and would be a good resource for you guys here in Wyoming with this technology. So just to outline how the process works a little bit, what you do is you make an agarose plug with the organism, a culture of the organism, and then you kind of rinse the plug and wash it so that you're removing the cell wall and the cellular debris, and what you're left with is kind of DNA in that plug. And then you cut that DNA, genomic DNA I should say, then you cut that DNA with what's called a restriction enzyme, which is an enzyme that cuts DNA at very specific sequences. And because these genomes are so large and it's just kind of random, it'll cut at a variety of places within the genome. And then you load the slices into a gel, electrophorese, and separate the DNA by size. And again, you stain with ethidium bromide and take a photo with a UV camera. And there's software that you can use to normalize the analysis. And there are also national databases. So I want to say that this is something that has been used for a long time for pathogens. There's national databases that contain thousands of images of human pathogens that have been isolated from real cases, probably millions, I don't know. I'll tell you about that process in just a second. But so what you're looking at here is, it's not the greatest gel. I'll show you. Wanda's was much prettier and more separated. This is actually one that I ran. So this was a sad story. It was like a Pseudomonas outbreak in a NICU, and they wanted to see if the Pseudomonas that was infecting the babies was the same as what was found in the sink. And so what you can see, these are a couple of different patients, and then these are a couple of different environmental isolates from the sink and the water. So one of the patients matched, you know, and the others didn't. So that's one way it's used in the real world. But this is the much bigger way that it's used in the real world, and this is PulseNet. It's what Wanda works on. This is a national network of people using PFGE to kind of track outbreaks. You know, you heard about the salmonella in the cucumbers. This is how they figured that out. PulseNet pathogens include everything that's listed here. Maybe what we can talk about sometime is salmonella or campylobacter. I don't know. It's interesting to think about looking for the pathogen in the watershed instead of, you know, just the indicator bacteria. I hadn't thought about that myself before, but this might be a way to do that. Because when I looked for information on PFGE and microbial source tracking, I couldn't find that much published information. It doesn't seem like it's not widely used for microbial source tracking. And maybe that's because it's library dependent, and getting a library together is a huge, colossal task, very time consuming. It would require just a large amount of hours. But I did see a couple of reports of E. coli and Enterococcus being used for microbial source tracking with this technology. So at our laboratory, we routinely perform PFGE for those PulseNet pathogens I showed you, not necessarily all of them, but a good number of them. And Wanda has, she does have a library of, I guess, non-pathogenic E. coli that were assembled from about 300 feedlot cattle. And I think that was in Colorado and Wyoming and maybe southern Canada. Sorry. Nebraska. So that library of the non-pathogenic E. coli does exist, but again, it's going to be watershed specific. There's a huge variation from what I understand within that those feedlots themselves. So it can be pretty complicated. But in thinking about this for the pathogen, it might make sense. I don't know. All right, I'm going to switch techniques again and talk about DNA sequencing. So DNA sequencing is the process of determining the order of nucleotide bases along a DNA strand. And you can sequence a single gene, multiple genes, or an entire genome. And there are new technologies that enable really kind of high quality, high speed, relatively low cost whole genome sequencing of cultured organisms at this point. So if you think about PFGE, it's providing a snapshot of the organism's genome based on where those restriction enzymes cut, but whole genome sequencing would provide you the exact genome sequence of the organism that you're talking about. So you might have heard the terms next generation sequencing. Next generation sequencing really is just an advance in a technology that makes sequencing faster, easier, cheaper, more applicable to kind of larger scale reactions. You can sequence whole genomes overnight. You can sequence multiple genomes from a single sample in one reaction. It's amazing what's being done with this technology. And so this is just an image of the instrument that we are about to get. It's called the MySeq, and basically the instrument itself is very expensive, and there are disposable kind of kits that come with the instrument that are also pretty expensive, but you can multiplex samples on that kit that make it much more realistic to use in terms of cost. So in terms of microbial source tracking, how have people been using next generation sequencing? Well, I've seen some reports, and they've been focused on, like I said, you can focus on a gene or multiple genes, specific regions. In some instances, libraries may already exist. So I'm gonna skip down here and talk about this paper. So basically, my summary thing said this shows promise, this technology, but it's still kind of an emerging technology, and protocols need to be standardized and things like that. But this paper was pretty cool in that what they did was they sequenced the genomes they created their own library, basically. So they took stool from various sources. This was in Japan. Various animal sources that they thought might be contributing to the pollution in it was a river, and human sources too. And I think they only sequenced maybe 22 whole genomes from these different sources. And then they compared those genomes to one another to look for regions that show host-specific differences. And they were able to identify some regions that showed nice correlation with particular hosts. And then they analyzed almost 500 samples that they had collected from that river using those host-specific sites that they had identified. And unfortunately, they were only able to link about half of them to a particular host species. The other half came up unknown. They couldn't be linked to any particular host. But that's probably because their library wasn't large enough, or because, like I said, it was a pretty small library. Or maybe some of the isolates just won't have any host-specific sequences. Either way, it was an interesting paper, and I think that you will be hearing a lot more about this technology in the future because it is the way of the future. In terms of our laboratory, we're getting the instrument within the month, and Wanda's already been to training on it, and we have another scientist who will be trained on it soon. We're going to be using it to kind of replace pulse field for those outbreak investigations and things like that. But we're interested in using it in additional ways as well, because like I said, it is pretty costly to use, and the more you fill up the little disposable reagents that they give you, the better. You can run a lot of samples on those. They call them chips at one time. So I think you've seen this slide before, but this is just sort of a broad overview of how you might use these techniques and where some of the techniques I mentioned would fit into the process. So of course you have your potential sources, you collect your samples, and then over here you have the library dependent methods where you're building your library from the fecal samples of your organisms. So over here would be something like PFGE. And then on the other side of the fence, you have the library independent methods where you're just extracting DNA from your sample and then looking for genetic markers specific to your host. And that would be like real-time PCR. And this is, I guess this is sort of my opinion based on what I have read of where where these things lie. So in terms of PCR and real-time PCR, the focus here is on a gene, right? And I've seen people target Bacteroides, Enterococcus, enteroviruses. This is not culture dependent, not library dependent. The discriminatory power is there, but it's not the best. The turnaround time's fast and it's pretty cheap. PFGE, the focus is more on a genome. I've seen people use E. coli and Enterococcus. It's culture dependent, it's library dependent. The discriminatory power is relatively high. The turnaround time is longer, about 5 days. And while the cost per sample is similar, oh, by the way, these are the costs that we would charge, not what a commercial laboratory would charge, because they're going to charge you a lot more than that. So these are like what they cost to run, I would say, not so much what you would be billed in the field. So even though the cost per sample is the same, you're going to need a lot more samples with PFGE just for building your library and things like that. And then next generation sequencing, it's almost like the possibilities are endless what you can do with that technology. You can focus on a gene, many genes, a genome. I've seen people look at E. coli, like in the Japanese study, they looked at E. coli. I've also seen community analysis where you're just looking at everything that's in that sample, every species of bacteria, and then trying to kind of correlate that with the gut flora of various animals. And I think that would be interesting but complicated. Let's see, and so because sequencing, there's so many different ways to use it, it can be culture dependent and library dependent or not. And the discriminatory power, if you're just sequencing a gene, is much lower than if you're sequencing an entire genome, which is probably the maximum discriminatory power you can get. And the turnaround time here would be a little longer, and it's going to be more expensive. So again, those are just my opinions. So do you guys have any questions?
When you have fecal coliform or manure patties, do they always have pathogens in it necessarily? Because I used to be a kid, and I followed agriculture, and I had to haul manure, and I was the ag waste specialist for Western Wyoming. But I fell into the manure one time up to my neck, and because the Stupid Ranch dairy farm tried to tell me that it was pretty deep. It never killed me.
Yeah.
And I've been around manure all my life. And that not all, just because you got manure, does not necessarily mean you got pathogen.
Right, I mean, same with you and me, right? Just because we poop doesn't mean we have salmonella.
Yeah. My neighbor got the other day.
So
He was eating some lettuce at some restaurant. But what I'm saying is, it just assume that you've got a pathogen because you've got fecal coliform. I think that's misleading to the public. Just because you got manure isn't going to be death to you.
no, I would agree with that. I mean, you know.
Do a lot of work for the vet lab also. And things that are not pathogenic to one species are pathogenic to another. And the big one in this state is Campylobacter. It's a commensal organism in cattle, but it's pathogenic to humans. And it can be pathogenic to cattle under stress situations.
So cattle can carry that with them, but I was thinking if a cow had a pathogen in itself, it'd probably die eventually, wouldn't it?
We have asymptomatic carriers of salmonella all the time.
Okay.
And plus, like what she said
And particularly
initially.
if you treat something with antibiotics, it can be an asymptomatic carrier for even longer.
Yeah. And like she said initially, you know, what's going to make you sick is different from what's going to make a cow sick. So.
Or make another person sick too, because I have an old rancher that used to eat meat that was black, and if I'd eaten that, he'd probably dead, but he ate it all the time. Bought it in one bit. So maybe he was an apathogen. What did you call that word?
An asymptomatic
Asymptomatic.
carrier.
Asymptomatic person. He told me that when I drank out of a cow with so many years, that I don't, nothing's going to kill me. And I really believe it. He lived to be about 90 years old.
Well, I would say that things like Campylobacter, your immunity is very short-lived.
Okay.
But salmonella can be fairly long. And I don't know if there's been a lot of, at one time they did a study at a health fair in Wyoming where they, you know, if you wanted to, you could give a stool sample. Or no, I think actually they took blood. And a university in Washington, I don't remember which state it was, screened for antibodies to E. coli 0157. And I think about 40% of that pool that were at the health fair that day had antibodies to 0157. So for those of us that are on well water or were raised on well water, yeah, we probably have some resistance to things like that now. But the second you're immunocompromised, it's an issue.
I mean, we did a study, and we wound up looking at healthy humans in a GI panel that looked for a huge host of different pathogens. And something like 30% of healthy people had Giardia. And that was collected around the Salt Lake area. So
I worked with a couple of studies, one back in the 90s and one about 10 years ago with Dr. Samapur out of the UW. And it was pulsed field. It was the methodology that he utilized. And in both of those, we utilized his library at that time. The first study, he had about 80,000 strains. The second study, about 10 years ago, we had about 160,000 strains. And then we collected samples locally in both studies, fecal samples, to compare to that. What are your just general thoughts about those types of libraries and the adequacy of utilizing them?
my first thought when you said those numbers was, whoa, if we needed to collect those, that's gigantic.
Pulsed field, or is it ribotyping?
It was ribotyping.
Yeah, I think that was ribotyping.
Well, maybe it was ribotyping. I thought he also used some pulsed field though.
That would be quite a few samples for pulse field. That would take a lifetime actually to generate that many. So that would be my first thought. If you had that library, that would be an amazing resource. Yeah.
But in terms of ribotyping then, and perhaps I was incorrect, what, again, the same question.
You know, I don't know that much about that technique, to be perfectly honest with you, and so I don't really have a good feel for what a decent sized library is with ribotyping. I just don't.
Okay, thank you.
Mm-hmm.
I live in Sublette County. We have a huge county, 4 different basically watersheds. If I have a beaver that's in the hoeback, is that E. coli going to be different than like in the green?
Potentially.
Is that what you said earlier?
Potentially, yeah.
And if I have 2 different streams within that watershed, will they be different as well?
So that is a question I would love to answer. And that, when we thought about doing PFGE for microbial source tracking purposes, sorry, I'm a little sick. We didn't know how to approach it, because with a human, you know, you culture the stool and you kind of assume that, well, you look for the pathogen. So you find the pathogenic strain amongst a whole huge variety of non-pathogenic E. coli strains. And so we don't really even know what that variability looks like, because we're looking at the pathogens. In the water, we were thinking that we would have to pulse every individual colony of E. coli that we pulled out of a single sample to understand the answer to that question.
Like I monitor 4 different watersheds. And, you know, it's like the hoeback goes into the Snake, goes into the Columbia, goes into the Pacific. Everything else goes into the Columbia River, or to the Colorado River. So just curious.
I'd like to help answer that.
Yes.
I can't fully answer your question, but I can tell you some of the things that we've found. So we do some pulse field gel on our environmental isolates of E. coli's that we find. We also do it on the pathogens that we find. And something that we have found is that there are certain strains of E. coli that seem to be ubiquitous. They're found everywhere, and they pop up consistently. And there are some populations of E. coli that if you did this kind of actual typing of E. coli, you would find that they're very clonal within a given area. So a population of E. coli is just replicating itself, so they all are sharing the same banding pattern that you're seeing in those gels right there. And then there are some instances where you'll see rare, different kinds of strains pop up in a population. So usually when we're doing our E. coli isolates is that we'll culture them out, and then we might take like 6 isolates and gel them out and figure out what's the level of variability within a given sample of that population of E. coli, to try to understand how could we determine if a source of E. coli was seen in multiple watersheds, or does it seem to be endemic to a particular system. And once again, I feel like you guys could all respond right here, it depends. But
That's okay. It's
so
no,
it depends. And connectivity between watersheds has a big role in those types of things because these things are moving through streams and moving downstreams. Mobility of the animals within that watershed matters also. If you've got highly migratory species that are moving through, that changes things. Cows that start off grazing, maybe downhill in the plains and stuff at one time of year and then are moved up in the mountains to do grazing, their gut microbiome will change and they'll pick up local strains of E. coli as they're grazing and they're consuming wildlife poop along with it. And they might get colonization of local types within there too. So you might be able to actually type one of your watersheds, or you might see seasonal trends in the types of E. coli that you're seeing there. But it doesn't mean that it's all chaos and you can't ever make sense out of it. Sometimes you do see clonal organisms in the same strains popping up time and time again.
Well, I think you had mentioned, or I can't remember if you said, something about sampling different times too. I mean,
Correct.
we don't do E. coli sampling, so I'm totally learning this. But you said, is it dependent on, like, after the spring of the year, I know I do water quality monitoring, like the chemistry, you'll see changes.
We were both saying is
what
a
you guys
huge
were-
seasonal dependence there. It really makes a difference. If you think about what we're like at different times of year, you're in the winter and you're eating all root vegetables versus the summer, you got salads all the time. Like you're changing, your gut microbiome is changing. The E. coli, when they're exposed to different things in the environment, depending on the time of year too, they're changing. Their biology is changing. They're putting out different kinds of enzymes. They're taking in little chunks of DNA through plasmids at this time of year, spitting them out at other times, encountering, antibiotics. They're evolving and changing all the time too.
Okay.
So yes, seasonal dependence. So getting, longitudinal data sets, you understand the trends in your data are very important because single samples, snapshot type samples, they can either be alarming or comforting, but they don't tell you about your system.
And then also, how about ambient or native bacteria in streams?
So that there is something to be said for that because we know that there can be native populations of bacteria out there, and some of the work I'm doing, you're talking about, actually doing community metagenomics. That's something that we do also to look at if there's actual invader, like phenotypes of bacteria, ones that are capable of like invading a native population and persisting, just like you'd have an invasive grass or something like that come in. So there's the potential for that also.
Okay, thank you, ladies.
Thank you. Other questions or thoughts?
Well, I just get an answer, so the E. coli bacteria is always changing,
It's evolving.
evolving. So when you would have a library one, you could go to another one in the future. You could have, would it always be the same?
That's
You
why libraries are
identify-
hard. That's why libraries are hard.
Yeah.
Some of you in here that have done some MST, do you have any questions or anything to add? I guess Tony or Dave?
The last one we
Teton?
did was in Teton County, Teton Conservation District. It was depicted earlier. And then the first one I worked with was in Ferry County Conservation District in north of Spokane, Washington. Actually, I contacted Virginia Tech and was directed to the UW, University of Washington, to conduct both of those studies. And we did a lot of background work and understanding. I mean, we were doing temperature modeling at the same time in the Washington study. We looked at riparian conditions. We did surveys of land use. There were much more extensive studies being conducted other than just looking at E. coli. We took water samples and we're looking at nutrient suites and other types of information as well. And we did a similar study when we conducted the one in Teton County. So I guess I would recommend that you really try to, as has been talked about here already, try to get a good handle on what's happening from a land use perspective. And if you have other information, that can be helpful too. One of the things that we're doing right now is we're working with our local county and we're developing some GIS layers, understanding, for example, where our septics are, that sort of thing. In Teton County and much of the West Bank and Jackson, we have water at or near the surface much of the year. So even if you've got a mound system, you're probably contributing to that situation there, the leachate from a bacterial perspective. So anyway, we're looking at a number of types of information is all I'm trying to get at here, to try to understand the bigger picture.
I'd say that we understand really well what the land use is, what the potential organisms contributing to the E. coli we've measured could be. And we did a reconnaissance sampling at each of our 4 listed sites just to see what the data looked like and get an idea what the results might look like. I would say to other districts who are apprehensive or landowners that are apprehensive about what they might find, I think in Wyoming the default assumption is that when you have a stream listed for E. coli, it is cows. And what we saw is, and what we knew, is that's probably not the case. So we have some data to start with, and now we can think about proper sample analysis plans and so on. And I'd mention for everyone who's curious that these 4 samples that we did for 4 potential contributors each cost $700 each at the lab in Laramie.
You had you mentioned SAPs, and this was something we were talking about earlier. For districts that have done like Teton, did you have a specific SAP for
We did.
MST
We had
sampling?
high QA/QC controls in place and an SAP plan in place before we started.
I would note in our reconnaissance sampling too, one of our sites where I wouldn't have been surprised to see cows, we didn't have a cow signal. And in a site where I wouldn't have expected to see much of a human signal, we had a human signal. And I was happy to hear mention of false positives and false negatives. That gave me stuff to think about.
Yeah.
Did you do any quantifying, or you just did straight samples?
The quantifying isn't really that quantitative.
That's what they were saying, so that's why I wondered where you went with that.
It might be quantitative within an organism is the way I understand, but with different organisms, you can't really say you get a number for cow and a number for human. You can't say that one is twice as much or 4 times as much.
I don't know if you've had a chance to talk about this. I was over in a totally boring meeting next door. But we've discussed the need to start sharing some of this information between the districts, including SAPs, costs, methods, disaster, train wrecks, whatever you've experienced, especially your experience with various labs, so that people that are just starting to get into this can hopefully skip some of the pitfalls. But also we really want to have a way that we can start assembling data, because as we go forward with a lot of these interactions with agencies, having good E. coli data available to universities and et cetera for them to work on is going to be really critical. So you're the point person for helpfully collecting some of this, and maybe even organize a workshop at some central place sometime in the future.
Yeah, that's where I was kind of wondering about the SAPs, and wondered, for those districts that are thinking about it or currently in or whatever, it's maybe sharing the development of that kind of SAP.
And the lab should be asking you that question too.
Yeah.
On their end, they should be asking you what kind of SAP you have in place.
We put together an SAP because something we were supposed to do, and now I felt like I pigeonholed myself too tightly, and now I want to retract that. Like I said, I was only going to do base flows. I was only going to sample when I had over 200 CFU per 100 mil of E. coli, and I went through the whole season without getting that opportunity pretty much. And discharge is really important. Flow regimes. Another thing is because it's so expensive to send a sample to a commercial lab, and it's not just the testing, it's also the shipping and figuring out the logistics. I had a sample arrive that didn't meet the temperature criteria, and so it's not real good data. I didn't have enough blue ice containers in there because I didn't get the. I was trying to save some money on shipping. It's a 48-hour holding time. So there's a lot of little fine details. I missed the getting it to the FedEx carrier once, and so there's that kind of stuff. I found that
So I think, maybe I'll sample on many hydrologic conditions. I mean, I could sample every week. I know what I was saying. Because I wait for my E. coli results, and it takes 24 hours. So let's say, well, I got 300 CFU. By the time I waited 24 hours and collected my sample for MST, and I also did another E. coli sample, turns out the next day it was only like 70 CFU. And I wasn't really concerned with a lower E. coli number. I wanted to know what the source was and a high E. coli number. So then what you have to do is, you're always collecting MST samples. Most of the labs I've talked to will take those samples, so they'll filter your water, a liter of water, through a filter and freeze it and hold it. They'll even hold it up to a year. So, if my board right now it says we only want to do human source identification, they're not comfortable with other targets. Well, when they come around and learn more information and are comfortable with testing other things, we can just go ahead and they've got the DNA and we'll test other targets. So, but that's expensive to ship water to, across the country, and you're still paying money, and it takes a lot of time and effort to do all that. So those are some of the little things, I'm finding.
We're about out of time, but we got a couple of minutes. DeMar, did you have something?
Real quick, are you going to have all their PowerPoints?
Yeah, I was just going to mention after questions that yes, we'll have the PowerPoints, and we'll have the video session with integrated power. Yeah, if the PowerPoint, we'll definitely have copies of that.
But just quickly, Kathy, my experience with this livestock problems in the fecal coliform from livestock basically is associated with these confined animal feeding operations that we have in our districts, where people are feeding cattle and they're having direct access to a creek or the fluid from their operation, their corrals and their units getting into the river. I cannot imagine that we can have a lot of problems up on our grazing lands, on Forest Service and BLM land from livestock type operations. I agree they get in there when they water, but it's these point source type, and a point source can be 6 or 4 cows, horses that have direct contact because they just denude the ground, and that's where it's getting in there. So you ought to, like you say, this reconnaissance deal, looking at what your districts are like and where you're picking up these high fecal coliform rates. Yeah, that's the biggest thing. You get those point source solutions. I consider confined animal feeding operation kind of like a point source problem. So be aware of that.
Yeah, Lucy and Melissa both alluded to know your watershed really well before taking on a. Yeah, Jill.
What is your goal? What are your monitoring objectives? And remember, E. coli is for human health protection. So look at your watersheds, monitor those reservoirs where the kids are getting in the water, or sections of your rivers or creeks that are going through town, but using it as a general just indicator watershed wide. I don't know if that's meeting your monitoring objectives. So it's always good to reevaluate why you're out there monitoring and really focus in on where you have human health concerns.
Yeah, anything else to add from DEQ?
I guess I would just add, we recognize these issues, and that in terms of setting standards, we know that there's issues with the E. coli standard that we have, the GME of 126. So we're learning about it too. I think Melissa, you said it, we need a place to start. That's what we have at this point. To change that standard is a pretty monumental task, and we're not opposed to that, but we're looking for solutions, and it's going to be a big change to find something different, something that's defensible, so that we as an agency are still meeting the requirements to maintain our delegated authority for the Clean Water Act program. So we understand the concerns. They're definitely important issues. We're open to discussing that, but it's going to be a long-term process to figure out how we would move to something that we feel is defensible.
I guess I'd like to add, as TMDL coordinator, I think the practices, when we're looking at a watershed plan or writing an implementation plan that's going to try to address E. coli, we get a lot of crossover between the practices. Things that are going to work for sedimentation and work for other things in the watershed that might have value, like wildlife. I've been attending quite a few of the meetings this last 2 days. There's different values that different districts are trying to target with their resource management. And E. coli might, we don't really want to talk about it too much, but the fact is the practice that will probably improve our E. coli concentrations, maybe not get us back into the water quality standards, but at least improve it, will also address other issues, other resource concerns. So I'd like to look at it more as a tool where we can kind of work together cooperatively and address E. coli, but also address other issues in the conservation districts that are important to the people that live and work in those watersheds. So kind of de-emphasize some of the E. coli, but emphasize the total ecosystem health.
All right, well, thank you, Alyssa, Lucy, and Sarah again, and we will also again get that MST white paper out this next week or so. Thank you, guys.